Hello everyone, I'm Mohammad and I'm here today with Luca to talk about our method, which on a very high level serves one of the fundamental questions in computational neuroscience, and that is, how do visual sensory neurons in our brain enable us to perform visual tasks like object recognition so well, and despite huge variations like in the background or lighting conditions, etc. And there are two core neural coding principles that support this. One is selectivity, which is where neurons respond to specific visual features, and the other is invariance, where they respond consistently across certain transformations. A good example for this is simple cells and complex cells in primary visual cortex, where simple cells are responsive to Gabor patterns at a specific orientation and phase, and complex cells, in contrast, are invariant to phase, meaning that as the phase changes, they remain highly activated. And here I'm using a GIF to show the variations in the phase, simply representing the invariance of the complex cell. Now, in our paper, we focused on invariances and specifically two questions. One was, how can we use a data-driven approach to characterize single neuron invariances? And the other was, once we have these single neuron invariances, how can we do a population-wide analysis to see if they organize in any way? And the challenges to do this, first of all, of course, in contrast to selectivity, where we are often interested in a single point in the image space, for invariances, we are interested in a whole set of images that may lie on non-trivial trajectories in the image space. And the other challenge is that there may be neurons that share invariance properties, have similar invariance properties, but the receptive fields have different spatial properties, meaning that they're located at different places, have different size or orientation. And to be able to compare them solely based on their invariances, it's important to kind of discard these differences that they have based on their receptive field. Now, our approach addresses these questions and challenges in 3 steps. In the first step, we learn the continuous invariance manifold of individual neurons using a generative model. And then once we have these single neuron invariances, we can compare 2 neurons by spatially aligning their invariances and see to what extent they activate each other. And then finally, we apply this on the whole population of neurons to see if we can cluster them based on shared invariance properties. Now, to learn the invariance manifold of a single neuron, we need a response predicting model that captures the functional property of that neuron, but importantly, we need this model so that we can optimize things on the input side using gradient descent. And as the image generative model that captures the continuous invariance manifold of the neuron, we use implicit neural representations, which are continuous functions of pixel coordinates. And we add this additional input dimension that allows us to generate multiple images using the same INR. Now, given a neuron, what we optimize are the parameters of the implicit neural representation to capture the invariance manifold. And the invariance manifold should fulfill 2 important criteria. One is that all the images that are generated by this, lie on this invariance manifold should maximally activate the neuron. And the other one is that the learned manifold should ideally span the complete underlying invariance manifold and capture its full diversity. And so the objective function that we use directly kind of relate to these 2 criteria. So we have a response maximizing objective on the left, and on the right we have a contrastive learning that encourages diversity by making images that are further apart based on how they correspond to the latent input to make them more less similar. And as a similarity metric, we use cosine similarity directly applied on the pixel values. So at this point, I'm assuming that we have applied this on a single neuron and we have captured its invariance manifold. And now we bring in another neuron. And visually, when we look at it, these 2 neurons have a similar invariance property. So if we take the implicit neural representation that captured the invariance manifold of the first neuron and show it to this second neuron, we would expect it to also maximally activate it, but it wouldn't, simply because their receptive fields are at different locations. But since we are using INRs, which are continuous functions of the pixel coordinates, we can apply a transformation on the pixel coordinates to spatially transform the learned invariance manifold while preserving the invariance transformation itself. And so in this case, the parameters that are being optimized are the parameters of this transformation, and we used an affine transformation here. And the objective is just maximizing the response, because we are not training the implicit neural representation anymore. And so we are only optimizing the affine transformation parameters. And that's basically the gist of the first 2 steps. And now I'm going to pass it over to Luca to talk about clustering and the results.
Thanks, Mohammad. Now that we know how to learn the invariance manifolds of single neurons, and we know how to compare invariance properties across pairs of neurons through alignment, let's suppose that we have a population of neurons. The question is, how can we divide it in groups of neurons that shared common invariance properties? The solution that we propose consists in basically applying the first 2 steps of our method to all possible combinations of neurons, store the activations that comes out of the alignment procedure in what it is basically an activation matrix, where each row represents how the invariance manifold of a neuron, in our case, in this case, neuron 1, activates other neurons after alignment. If 2 neurons share the same invariance property, then they will be highly activated. So the invariance matrix might look a little bit like this when we have a larger population of neurons. And as it captures functional similarities across neural encoding across neurons, we can cluster it using a clustering algorithm, such as, for instance, agglomerative hierarchical clustering. And in this way, we can find groups of neurons that share common invariances. Let's now look at some results. First, we wanted to validate our method in a control scenario. So we tested it on a population of simulated neurons. We created 6 different types of simulated neurons with known ground truth invariance manifolds. So we have simple cells, complex cells, and then 3 additional types of neurons that presents more complicated arbitrary invariance manifolds. For each of these neuron types, we created 6 different instances that relate to each other by an affine transformation of their receptive fields. In this way, we obtain a neuron population that might mimic the variability that could be present in a population of neurons from an early visual area of the brain. We apply our method to learn the invariance manifolds of these neurons, and as you can see here in the bottom, these are the learned invariance manifolds, and the images in these invariance manifolds closely resemble the images in the ground truth invariance manifolds above. When we align these invariance manifolds through all the pairs of neurons and apply hierarchical clusterings, we are able to reconstruct the 6 different groups of neurons that were present in our neuron population. Okay, happy of our results on simulated data, we then move to apply our method on biological data. In this case, we use a response predictive models that was trained on macaque V1 neurons data. The data set was collected from Andrea Stolia's lab and contained responses to grayscale natural images. We trained our model and selected the top 100 best predicted cells. We applied our method and identified in this way 5 different clusters of neurons. The largest 2 of which represents, as we expected, the known functional cell types of simple cells and complex cells present in the primary visual cortex of macaques. However, remarkably, we also identified 3 additional clusters of cells that presents invariance properties that were previously not described in literature. And neurons in this cluster seems to be selected to more complicated types of stimuli, of features in the stimuli. So for instance, neurons in cluster 5 seems to be selected to patterns that resemble a little bit more checkerboards. Okay, these findings applies to the neurons in the response predictive models. Does it actually generalize to their biological counterpart? Now, this could be tested with neuroscientific experiment. This could be tested definitively. However, this possibility was not possible for us. However, we have evidence to believe that shows that this is the case. So first of all the neurons that we considered are very highly activated by our response predictive models. Second, we trained a second model, a different response predictive architecture, a second response predictive model with a different architecture on the same data set. We show the invariance manifolds of the first model to the second models, and we confirmed that they were still very highly activating. And third, our population of neurons was collected from 2 different macaques, and in each of the clusters we identified, there were neurons belonging to both animals. So, in conclusion, we presented a method to study invariances in population of biological neurons. We call this method laminar because it learns invariance manifolds and aligns them across population of neurons using INRs. When we and through laminar we can generate an activation matrix that we can use for neuron clustering based on shared invariance properties. When we apply laminar on a population of macaque V1 neurons, we identify simple and complex cells, but also additional clusters corresponding to neuron presenting novel invariances. If you're interested in laminar, you should check out our GitHub repository. And with this, I would like to conclude. Thank our supervisors, Jan Antolík and Fabian Sinze, also from Mohammad. And I would like to thank you for your attention. And we would like to invite you to talk more at our poster.